Welcome back. Today we're digging into some really fascinating research that honestly flips the script on how we think about successful cancer immunotherapy.
It really does.
The big takeaway, the sort of counterintuitive point. Predicting if immunotherapy works isn't just about counting killer T cells. It's about where they are and what they're doing, specifically the geometry of it all.
Exactly. We've spent ages, labeling tumors hot or cold based on bulk T cell numbers.
Right, just a simple count.
Yeah, and it's kind of like judging a whole city's economy just by its total population. You completely miss where the actual interactions, the real business, is taking place.
So this paper maps that action. It
Yeah.
identifies these specialized little zones, these micro niches, where T cells actually meet and, crucially, kill cancer cells. They call them craters.
Craters, cancer regions of antigen presentation and T cell engagement and retention. It's quite a mouthful, but descriptive.
It is. And that definition is key. So our goal here is to unpack how these craters form, what makes them effective kill zones, and, importantly, why their density after treatment seems to be a powerful predictor of whether a patient benefits.
Especially with immunotherapy checkpoint blockade, or ICB, that connection is vital.
Okay, let's start with figure 1. They kicked off with a pretty basic question. Where do the T cells actually interact with tumor cells?
To answer that, they used live 3D imaging, which is pretty cool in itself. They used a zebrafish melanoma model.
You're giving them a real-time view.
Exactly. And the result was clear. The CD8 plus T cells, the killer cells, weren't just scattered randomly. They clustered in these specific pockets, the craters.
And where were these craters located?
Right at the boundary. The interface between the melanoma cells and the surrounding tissue, the stroma, and the T cell density there. Significantly higher than elsewhere. This wasn't random chance, it was targeted.
Okay, so location matters.
Yeah.
Geometry is key. But figure 2 takes it a step further, right? It asks why they stay there. What holds them in these craters?
Is it just like a sticky spot or is something more active going on?
Yeah.
Well, it turns out it's active chemistry. They used spatial transcriptomics, looking at gene activity in specific locations, and found these craters were packed with B2M,
Beta-2 microglobulin.
which is part
It's
of...
a core piece of MHC class I. That's the machinery cancer cells use to display antigens, like little flags saying, I'm here, come get me to the T cells.
Okay. So it's not just a physical niche, it's an active signaling hub. The T cells are being told to stay there because the antigen flag is waving strongly.
Precisely. It's active recognition. And they proved it. They used CRISPR to knock out the B2M genes, specifically in the melanoma cells.
Then what happened?
The T cells didn't stick around in the craters nearly as much. The ratio of T cells in craters versus elsewhere dropped significantly. It showed that stable retention really depends on that active antigen recognition. No flag, no T cell engagement. Okay, that makes sense. So if craters are where the killing happens, the functional hotspots, here's a question for you and for everyone listening. If immunotherapy is working well, should we expect these craters to shrink as the tumor dies, or should they actually expand?
That's the critical question. And the answer, if they represent an ongoing, effective immune response,
Is that they should expand.
they should expand. And that leads us right into the human data in Figure 7. They looked at biopsies from human melanoma patients after they'd received ICB treatment.
And they measured what they called crater linear density. Basically, how many craters per millimeter along the tumor edge.
Yep, comparing patients who responded to the therapy versus those who didn't.
And the result.
Clear difference. Patients who responded had a significantly higher density of these craters in their post-treatment biopsies. It wasn't that the craters disappeared, they became more numerous in responders.
Wow. So more craters equals a better sustained anti-tumor response.
Yeah.
That's a powerful clinical link.
Absolutely. And that's the real innovation here. Crater density isn't just another bulk metric like counting T cells or looking at PD-L1 expression, which can be, unreliable.
Right. It's spatially resolved, it's imageable, and it's directly linked to the function we care about, the killing.
Which leads to some exciting implications. They talk about niche amplification.
What do they mean by that?
Well, instead of just trying to boost T cell numbers or activity systemically, maybe future therapies could focus on creating or expanding these crater niches directly within the tumor.
How would you even do that?
Maybe by editing the extracellular matrix, that structural scaffolding,
No.
or reprogramming the stromal cells to be more supportive of antigen presentation and T cell retention?
So actively engineering the tumor microenvironment to make it more conducive to killing. Turning cold tumors hot by fixing the battleground.
Exactly, fixing the microenvironment itself.
Okay, let's quickly recap the 3 big takeaways then. First, effective tumor killing isn't random. It happens in specific microscopic zones called craters.
Second, these craters work because they're rich in B2M and MHC class I, actively presenting antigens and retaining the killer T cells.
And third, crucially, seeing more craters after immunotherapy is a strong sign that treatment is working well in patients.
There is a key limitation, of course. This needs standardization. We need bigger prospective trials to really nail down how to measure crater density reliably and confirm its predictive value, maybe using biopsies taken early in treatment.
Makes sense. It needs to be robust before becoming a standard tool. So final thought here. They mentioned seeing similar structures in lung cancer too. Could this crater concept be like a universal feature, a common kill zone architecture across different solid tumors?
That's the really provocative idea, isn't it? If we could define this universal structure, maybe targeting or creating it could be a broadly applicable strategy in cancer immunotherapy, something definitely worth exploring.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.